Yes, sir.
So do you follow it? Are you able to understand it? Hello? Hello, students?
Yes, sir.
Yes, sir.
Yes, sir.
So why you are taking this much time, mister? You are able to understand, right?
Yes, sir.
Okay, so I will send you some more files and I will explain those experiments. Okay, so what you have to do is, so just follow that. Okay, put your name and make it like exercise 1, 2, 3 and so on. Alright, and then you prepare like a record in your name. Okay, each of you should prepare the record and finally, like when we complete all the exercise, like you have to send through email. Okay, so keeping that as your like internal, I will give you the marks. Okay, so all of you should submit the record like once like all the experiment is like over. Okay, so I gave you some exercise, right? All of you should visit like PDB site and I asked you to do some exercise, right? So did anyone try that?
Yes, sir.
Yes.
Yes, sir.
Yes, sir.
So what you did?
We downloaded the sequence file from the PDB, the 10 sequence. You said that.
So in a batch mode, all right?
Yes, sir. Yes, sir. In
Yes, sir.
batch mode, and we can see the coordination of the atom in that file.
Okay, so you downloaded coordinates as well as the sequence.
Yes, sir.
Okay, so did you compare the proteins?
Yes, sir. Structure similarity we have done.
Yeah, structure similarity, how you did?
Sir, in PDB we have an option called Advanced Search Query Builder, through which we can key in the PDB ID under structure similarity. After that, we can search for the. There are many options available for the display. Like we can see only the similar PDB IDs, or we can even go for the entire details of it.
Okay, that's good. Okay, so this exercise you have to make your own statement. Okay, you have to make aim and give the title as like exploration of like PDB site. Okay, and put like aim and procedure like step by step all you have to write. Okay, and then take a screenshot of your result. Okay, take a screenshot of your result and give it in your record book. All right.
Okay, sir.
Yes, sir.
Okay, sir.
Okay. So all of you, do you have PyMol software with you?
Yes, sir.
Yes, sir. I have.
Yes, sir.
No, sir.
No, sir.
No, sir.
Why no? Those guys saying why no, why you are saying no? The PyMol is free software that you can download it.
Yes, sir. I will download it.
Okay. You just download in your PC or in your laptop and you can learn it. All right. So all of you are familiar with PyMol. Is anybody not having any experience in PyMol? You can say. That means everybody is like already familiar with like PyMart. Okay. Okay, then what I am going to do is, like today I am going to talk about structure comparison or like a structure alignment. So we know like a sequence alignment, right? So sequence alignment, what you will do? So you will take like a two sequence and like a character by character, you will do the alignment, right? Character by character, you will do what? Comparison, right? So from the N terminal to C terminal, each character you will see whether they are same or not. If they are same, you will leave it. If it is not same, so you will try to give some gap, and so that you will try to have like a better alignment. So that is like sequence alignment. Okay. In case of protein structure, how will you do? So what is the idea behind like protein structure comparison? Okay, that is what we want to do. So last week I asked you to do like a structure comparison. So some of you, I do not know how many of you like did that. Like the Subhas replied me. So I believe like everybody done. Okay. So in PDB side, so you have selected like a similar structure and you did like a structure comparison. How is this actually done? What is the idea behind that? So that's what I'm going to see, discuss it right now. Okay. So if you go to the PDB side, so once you downloaded the PDB file, you will see the like the PDB looking like this. Okay. So initially you will see the header section, header and instruction about the like a particular PDB. Okay. So here you will see like the title card. Okay. The title card says like a protein name and like protein published the title which was where it was like published. Okay. And the compound name and the source from where you got the protein. Okay. All like detail, whether like experimental detail also, experiment data, whether it is like X-ray diffraction or through NMR, whatever. So all the details that you will see, and journal, the reference also it is given. Okay, in which journal it was published, was the title. Almost this title and this title will be same. Okay, in some case they may be different. It depends upon the author who is submitting the PDB. Okay. So you will see the header until you reach the PDB coordinates. So the PDB file, it is having like a specific coordinate where you can see like here the usual format represented here. Here in coordinates, like you have like two sections. One part is representing for the protein or DNA, and another part is for heteroatom. Heteroatom is for heteroatom. Okay. Maybe for ligand or ion or like water molecule. So all those things are denoted by het atom. Okay. So for protein or like a DNA molecule, each atom it will be starting with like a code like atom. Okay. And then you have like an atom number. So here you can see like after atom 4 letter you have like a space. Okay. This space, so you can it can be utilized for again like if you have like head atom something like that, you can use this space. Okay, and then this space is like also utilized by the atom number. So here you see like 1, 2, 3, 4, 5 and so on. Then it is going like 2000 more, right? 2000, 3000, something like that. So this space will be like still utilized. Okay, so it is having its own format. Okay, it's not just like a random given. Okay, it is following the certain format. Okay, so atom number and atom identity. Here you can see like Ca. So Ca, C. What is this? Both are carbon only, but Ca is alpha carbon, C alpha, right? In a peptide, you have like a C alpha atom and C prime atom. C prime is what? A carbon which is connected with a double bond O. Okay, so like a C prime. So 2 carbon you have in each peptide. So to differentiate it, so it is like alpha carbon where you have like a side chain attached. This is C prime atom and oxygen, nitrogen, C alpha atom, C prime, oxygen, C beta. This is like a side chain. C beta, C gamma, C delta, O epsilon 1, O epsilon 2. Okay, if you have like multiple, so if you see the glutamine, you will see like all these atoms, right? So glycine only like 4 atoms you have, right? So alanine, you have like N, C alpha, C, O, and C beta in alanine. Okay. So depending upon the residue, the atom identity will also like vary. So here you can see like the residue name, residue identity, the chain name. So in some protein you will have like a multiple polypeptides. So each polypeptide given in a different chain. Okay. So A chain and B chain, so on. So residue number. Okay. And then you have like the coordinates like X, Y, Z coordinates. Okay. So here are like some crystallographic parameters. So you don't need to worry about this. Okay. So this is like a general PDF format that you might have seen in the PDF file. So now we are going to see this like a structure-based like alignment. How this like a structure-based alignment we are doing. Students, you are able to follow, right?
Yes, sir.
Yes, sir.
Yes.
So here in structure, again you can try for like a global as well as the local alignment. Okay. So I have like some proteins just like this. Okay. So when I want to when I am asking you to compare these two proteins, what you will do? You will try to do the superimposition. Okay. So take this protein, keep not moving this one, and you try to move this. Okay. So when you open these two proteins, it may be in like a different coordinate. Okay. So what you will do? You will try to bring this protein close to this and try to do the rotation. Okay. And then what you will do? Then you will try to overlap on each other. Okay. So that is what like a structure alignment, right? That's what like a structure comparison. So when you do like a superposition, when you do the structure alignment, you can easily say whether both structures are like aligned well or not. Okay. So here you can see like a structural identity, it is like a 90%, but like sequence identity is like just like 27%. Okay. So the structurally it is like similar and it may carry same function. So such a like biological decision that we can make by doing like a structure alignment. So how can we do? How can we make this superimposition? So assume that you have like two molecules just like this, right? So what I have to do is, as I told you already, you have to identify the parts, right? The parts which has to be overlaid. Okay. So I have to bring the head to this head here, bring the tail to here, right? I cannot do like a reverse. I cannot bring like head here and the tail here. That is not like the best superimposition, right? So which part has to come, that I have to design clearly. Okay. So according to that, I have to do the transformation. Okay. So even though like I have like transform, okay, still it is not matching. The head is somewhere away from here and the tail is also away from here. So I have to do some rotation. Okay. So I have to do some rotation so that all the parts are like overlying on each other like nearly. Okay. So for this, I need to have like three kinds of like transformation. So I have to do like a rotation, or I have to do like a translation, even scaling I have to do. So translation we can understand, and if this is a molecule like this, okay, once you done with your translation, then you have to do translation. translation, and then you have to do rotation. Okay. In some case, you have done like a translation rotation, but the origin is different. So you have to do the scaling, right? So you have to move it close to each other, right? So you have to do the scaling. So all this transformation that generally you have to do in order to do the overlapping. Okay. So here, like you see, like two configuration of like points. Okay, I am asking you to do the like a superimposition. So what you will do, you will try to do like all the possibility. Okay, you will try to do like all the possibility. So you're trying to align like this, or trying to align like this, or trying to align like this. Okay, so which will be good? So this looks like a much more better than this, right? So you can understand like best like transformation. Okay, so all these are like just like a different example that in order to make you understand like how you are doing like a superimposition. Okay, so now I have done the superimposition. Okay, how far my superimposition is correct? Okay, so in case of like as I told you, like a sequence alignment, what you will do, you will give some scoring, right? Out of 500 amino acid, right? So say for example, like 400 amino acids are aligning very well. So what you will do, how will you calculate the score? 400 divided by 500 into multiplied by 100. Okay, so that will give you like something like a 90%, 80%, whatever, right? So maybe like only 100 amino acids, like superimposing, 100 amino acids are aligning out of 500. What you will do? 100 divided by 500 into 100, right? So maybe it will come like only 15%, 20%, something, right? So based on that, you are giving some scoring. So here, how will you give the score? When you are like superimposing, how will you give the score? So here, the score is given in terms of root mean square deviation, RMSD. Okay, root mean square deviation. So here, so you have like a protein A, you have like a protein B. So you have like a 1 to n amino acid. Here 1 to n amino acid. I can say in terms of like a two sets of points. So A protein, where I have the points like I is equal to 1 to m, and B protein J is equal to 1 to m. So pairwise we are doing the correspondence. Okay, so AK1, BT1, AK2, BT2, and so on. Okay, so that's why you are doing the RMSD. So square root of summation over AKI minus BTI whole square divided by n. Okay, so that will give you the RMSD, root mean square deviation. So when you are calculating the root mean square deviation, so you can easily say like how far my protein is like aligned with the target protein. Okay, so you will get like 1 angstrom RMSD, 2 angstrom, 3 angstrom, 4 angstrom, or 5, 6, or 23, 50, whatever it is. Okay, so suppose I have like 23 angstrom. What does it mean? 23 angstrom means the protein is not at all overlapping. What about like 50 angstrom? Definitely it is not overlapping. Okay, so the RMS deviation is what? It has to be low in number. RMS deviation, the deviation should be very, very less, which represents the both protein are very well aligned. If the deviation is like too much, the alignment is not good. So that is what you have to understand. Okay. So the alignment may be like anything, like a rigid alignment or like a flexible alignment right? So assume that you have like a protein. So here you can see this both are same protein. Okay. So if you see this one, it is in an extended form. You have like one domain here, another domain here. Both domain is connected by a long alpha helix. Okay. Whereas this protein, you can see these two domains have come together, right? So this domain has come together. It's getting like a closed conformation. It is because if you see here, the brown color, right? The brown color is a ligand, right? It's a ligand. So once the ligand is binding here, so this domain, right, is going towards here, and the ligand is occupying this space. Okay, that is why it is a conformational change. Students, do you follow?
Yes, sir.
Yes, sir.
Okay. Same like this is A domain, and this as like a B domain. So here also you will have the same naming, right? So you will have like A domain here, and you will have your B domain here. But only thing is the conformation is changed now. Okay, it is because the B domain is moved close to A domain. So the conformation is changed. Okay, so this is a free form. This is like open configuration. This is closed configuration where you have like a ligand. Okay, so now I want to have the alignment between like open configuration and closed configuration. So that is what I am trying here. So what it will do? It will try to align either with A domain or with B domain. So say it is aligning with A domain. So what you will get? So only this domain will be aligning here. That is what you are seeing here. And the B domain is not aligned. Right? So the RMS deviation will be what? What will be the RMS deviation? Here the RMS deviation will be very high because most part of the protein is unaligned. The most part of the protein is unaligned. Only like 50% of like protein. That is only one domain of the protein, entire protein is aligned here. Okay, so the deviation will be too much. Alright, but if you look out for only this domain, the alignment will be very good. The deviation will be very, very less. Now, students, you follow?
Yes, sir.
Yes, sir.
Okay, so if you look out only this domain, the deviation will be very less. Similarly, if I look out deviation only for B domain, if I align only the B domain here, the deviation will be very less. Alright, so this concept, this idea, you should understand. Okay, so here basically the RMS deviation, this formula. Okay, this formula is very important. Okay, right? Either you can write in this way or you can write in this way, whatever it is. Okay, so all right, whatever. So that's all. So this like basic thing about like structure alignment. Okay, so I'm going to share you the PyMol screen here. All right, just wait for a moment. I don't know whether I can share PyMol. I guess no, it's not coming. Are you able to see? only the console is coming.
Yes, sir.
But do you see the protein screen?
Sir, for that you need to share your full screen, so then we can see it.
No, this window is not appearing. I don't know.
No, sir. After clicking on the presentation button, presentation tab, you will see 3 options: your entire screen, a window, or a tab. So you have to click on entire screen, then you can share it.
No, PowerPoint is final screen. That is not coming. Sharing, I should see that now. So even if I put like entire window, it is not showing the that viewer, PyMod viewer. It is not coming. It is not appearing, even though if I share like entire window, it is not appearing. Only that console, PyMod console is coming.
Yes, now we can see that.
PyMod window you are seeing. Do you see the PyMod viewer?
Yes, sir.
Yes, sir. Yes, sir. Yeah.
Just hold on. So here, if you are able to see this, right? So here, I have like two programs. Okay. So how can I do that? Like, I want to show you a demonstration of like superimposition. Okay. So here I have like one protein, like here I have like another protein, right? So how can I? Do you know anybody? How can I do the transformation here? Or superposition here in PyMod? Well, okay. So here you can see like the action button. Okay. So go to action, and if you see here, it is showing align. Okay. So align to molecule. Okay. You have to select the molecule. All right. So what I am doing is I am keeping this like as a still object. I am not moving, but I am moving this object. Okay. I am moving this object. So what I am doing is going action to this protein, go to the align to molecule, and the other molecule will be displayed here, and you are done. Okay. So if you see here, the RMS deviation, it is showing 16.44 angstrom, 16 angstrom. Okay, so 16 angstrom means what? It is not aligning. Okay, so what I will do is you can lines. So if you see here, if you see the secondary structure, students were able to see, right?
Yes, sir.
Yes, sir.
Yes, sir.
If you see here, the most part of the protein is not aligned. Okay, so that is why you are getting like a 16 angstrom. Okay, if it is like a good alignment, the what is it? The deviation will be very less. All right. So pi mod you can do like alignment. All right. So you go to the axon. So there is like so many options are there. Okay. So you can have like a different kinds of representation. Okay. So I'm representing in a cartoon mode here, right? You can just represent like a sticks, ball and sticks you can do, or you can do the surface representation, right? So what I will do is I will hide the streaks, okay, hide surface, okay. So what I am going to do is I can do the surface based on charge as well. So for that you have to go to like axon, okay, and then generate like vacuum electrostatic. Okay, go to generate, go to vacuum electrostatistics, and select protein contact potential. Okay, so once you done, so you might have seen it was calculating immediately. Once it is done, okay, so it will show you the electrostatic potential surface, okay, charge surface. So here the charge surface, you can see like a blue color is a positive charge, red color negative charge, and the white patches, it's indicating like a neutral or like hydrophobic environment. Okay, so you can see like the charge potential scale also is there, right? So you can like represent like any kind of way that you want to. Okay, so you have to practice like pi mod, and you have to learn like each commands here. Okay, so this is just a demo. I cannot like give like all these things. Okay, so try yourself, and if you have like any doubts, you can ask me. Okay. So I have told about the pi mod, right? So if you want to do like a superimposition, if you want to have like a structure alignment, okay, there are different servers available. Okay, for example, VAST. Okay, VAST. VAST means vector alignment scale 2. Okay, vector alignment search tool. Okay, so you can go to the VAST site, and you can do the structure alignment. Similarly, there is another server called like a DALI. Okay, so DALI protein structure comparison server. Okay, so both server I will give you a demonstration so you can follow that. Okay. Right. I think the screen is visible to you, right? You are able to see the screen, right?
Yes, sir.
Okay. So here is the DALI server. Okay, the screen showing you. So here you can see this, like there are like different options there. So daily what it will do is just like a blast. In a blast, what you will do, you will give the protein sequence and it will go to the database and it will bring you similar protein sequences. So just like that, daily if you supply your PDB, so if you supply your PDB, it will go to the database and it will do the comparison. It will do the comparison and it will bring you all the protein structure that are very close to your protein structure. Okay. So the daily will not do the sequence alignment. It will do only structure alignment. Alright, so you have to remember this. So you can give one PDB and you can ask for the find out the structure, entire structure, all the structures that are close to yours from the database that you can do, or you can give your protein and you can specify only 10 protein structures to be compared, or only 25 structures to be compared, whatever that you can do. Okay, so different option is there. PDB search, like PDB 25, pairwise search, whatever, like different search. Okay, so go to this site. Okay, try to learn all these options. Okay, so what I'm doing is like I'm selecting like a PDB search. So once you done like PDB search, it will ask for the query protein. So here you have to give your protein PDB ID, or you can select file also. Students, like I'm clicking all this window, I assume it is appearing in your screen.
Yes, sir. It is appearing, sir.
So tell me, because I do not know. Okay, so you can select your file, choose file, and select any PDB ID that is in your local system, and then you can give like a job ID, okay, and you can give it like email ID and then submit. Just submit it. So daily it will do like a very good calculation, okay, so that it is going to take a longer time. Okay, so this is like PDB search, like PDB 25. So it will take longer time, and once it is done, it will give you the result just like this. Okay, so here you can see matches against like PDB 25, matches against PDB 50. That is, it is comparing with like 50 proteins, comparing with 90 proteins, comparing with like full protein PDB. Okay, so 25, the result will be very limited. You can like compare very easily. So I'm just clicking this like PDB 25. So here you can see like best proteins, okay, which are like aligned here. So here you can see it is showing 3kZH because I selected, I have given the protein as like 3kZH. So this is my input, my query protein. Okay, so it will do 3kZH. It is available in PDB also. So it will do that protein also, comparison with that protein also. So RMS deviation naturally 0 because both protein will be aligning same, right? So local alignment, the entire 100% identity it is showing. It is like your protein only. Then second you can see, okay, it is showing RMS deviation 2.7 angstrom. Alright, so here Z is given. So Z represents the score here. So that has to be very good. Okay, 32.6. Okay, so here it is saying RMS deviation is like 2.7. Okay, so I am looking for the Z score. So far 32 is the best. So I am going down. So here I see like 26, 27, 28, and not these scores are like very less. 2.13. This is like very less. So I feel like this one, the second second in the list is like a very good alignment. Okay, so this is like a carbohydrate kinase, and the orange deviation is also like somewhat like reasonable, 2.7. Okay, 2.7 is like very high, but in this one, in this search, it looks like a very good. If you see like any other protein less than 2.3, I don't see like any like very less RMST. So everything is like more than 2.7. Here like another this is another carbohydrate kinase. So here also carbohydrate kinase. So again like 2.7 angstrom. Okay. So this is how you will do the alignment. Okay. So here it is showing the sequence of like query and the subject as well. Okay. So here it is listing like the all the sequences. But the thing is, if you note down, okay, so it is showing LLEE. What is this? So this is loop region. L represents loop region. E represents secondary structure, beta sheet. Okay. H represents helical region. Okay. So which helical part it is matching? So the first protein is what? First protein is same protein. So you will see all the residues are exactly matching. Okay. So don't be disappointed by seeing the first protein here. Okay. But if you see the second protein, you will see like a lot of variation in sequence also. But the thing is, you will see the secondary structure like somewhat very close. Okay. So that is what the advantage of like using the DALI server. Okay. So if I want to have like a three-dimensional viewer, okay, so just click this through 3D superimposition, right? So it will show you your PDB. Okay. So it will show you your PDB. I have not selected any PDB here, so it is showing only my input. Okay, so what I am doing is I am selecting my protein. Okay, so I am selecting my protein, and again I am doing like a 3D superimposition. So now it is showing the aligned region. Okay, students, you are able to see this, right?
Yes, sir.
Yes, sir.
Okay. So initially I did not select this, so it was showing only my query protein. So now I have selected this. That means the DALI will understand I want to superimpose this with my query protein. Okay, so it is aligning and it is showing like superimposition of both proteins. Okay, so here you can see like most part is like aligned well, right? So this is how you can use like this DALI server. All right. So here you can see like the sequence identity is like very less. All right. Okay. So what you can do is go to the DALI server and take any PDB ID and submit to the DALI server. Okay, find out like the result here. Where is it? Yeah. So the result, the previous page. Okay. So it was showing you. Yeah. Yes. So in the previous page, it was showing you like PDB 25, PDB 50, PDB 90, like that. It was showing. So you take a screenshot of that page, and then I selected only the first one, PDB 25. So it was listing me this thing, okay, this page. All right. So you have to take a screenshot of this and try to make alignment of like two or three proteins, just like 2 or 3 proteins, and make a screenshot of the superimposition screen. Okay, so I have not explained the algorithm behind the DALI. Okay, the idea is same. I told you the formula already to find out the RMS deviation. So what it will do is, it will take the first residue of this protein. Okay, first residue of this protein and the first residue of this protein. So it will do the calculation of RMSD, and then it will take the second residue and second residue here. Okay, here you can see this the length is very short. In a subject protein, this part is missing, so it will not worry about this. Okay, so that's why you are getting like a RMS deviation too much. Okay, so in this position, the RMS it will try to do the alignment. Okay, all right. So, and I told you another server, VAST, VAST. Okay, Vector Alignment Search Tool. Okay, so here also you can submit your PDB and you can find out like a structurally similar protein. So here you can see like so similar structures for same PDB ID. I have just select this and hit go. Okay, so here, so you will get a list of result of like this. So here it is telling you like 2,800 selected structures. Okay, so once I have given like a 3KZH, it was searching the PDB site and it is selecting nearly 2,900 proteins, and it is showing the result here. Okay, so here you can see the RMS deviation for all these things, like it is like showing like 6.19 angstrom, like more, almost like 6 angstrom, like 4 angstrom. Okay, so how many residues are aligned and sequence identity is what? Okay, so all these things it is listed here. So if you want to list more than 10, so here you can increase the number. Okay, or else you can go to the next page, or go to the last page, whatever. Okay, so you can do, and so what I will do is you can see this positive sign. Just click this, so it will expand for this protein. Okay, so now it is showing me 3KZ and comparison with this protein. So the A is my query protein, B is like subject protein. It is aligned with B. Okay, 6 ILR. Okay, so if you want to the sequence alignment, you can just click this. A pop-up window will come. Okay, so it will show you the. You are able to see this, students. This window,
yes, sir.
Sequence alignment window. Do you see?
Sir, we can see that first.
Okay. So it is popping up in another window, so you may not see. So I will show you here. So you can visualize 3D structure superimposition. Just click this one, so it will appear in this window. Okay, now you are seeing this window, right?
Yes, sir.
Okay, so here you can see, like both proteins are like aligned. Okay, so just to move the cursor, it will show you the residue name, PDB ID. So the blue color is the query sequence, our protein. The red one is subject protein. Okay, so here you can see the sequence also like displayed in an alignment fashion. You can see like a beta sheet region which are like aligned. Okay, So like loop region, helical region, everything is like displayed with which is representing like a secondary structures. Okay. So this is the VAST server. Okay. So what I will do is you can take like some PDB and you can again try with VAST site. Okay. So you have to try like both like daily server as well as the VAST server, and again you have to make the exercise like result, all those things that you have to make on your own. Okay, and you make like everything and send the soft copy to me. Okay, I will make corrections and then you can make it as like a record. Okay, so now so far like we have seen like a structure alignment. Okay, what I will do is I will so we have experiment of like estimation of DNA. Okay, so I will give you some solution and I am asking you to find out like how much amount of DNA is present. Okay, by means of by doing the experiment. What will you do? So there is an experiment so where you can use the diphenylamine. Okay, so you will take DNA and it will interact with the diphenylamine so that the color will be changing. Okay, so the color will be changing. So how far it is changing, that you can study by using the UV spectrophotometer. Okay, so what I will do is I will send you the file. Okay, so it is explained in detail. You can read that. It is self-explanatory. All right. So there is another experiment. This is again like a melting temperature of DNA, finding melting temperature of DNA. So what is this? So we know like DNA is like a double stranded. Okay, so how can I find out the melting temperature? So I have to heat the protein. So if I heat the protein, what will happen? The double strand will become single stranded. So I have to keep on heating. The temperature should be keep on increasing. At one particular point, the DNA will become single stranded. Okay, so now whether it is still in a double stranded or like a single stranded, how can I find single stranded? How can I find it? So by using UV spectrophotometer, I can find it. Okay, if it is a double stranded, it will show me some value, right? Some optical density, right? So if it is a double stranded, again it will show me some OD, right? So based on this difference, I can find out whether DNA is single stranded or double stranded. Okay, so usually when the double stranded DNA is there, the UV reading will be very less. Whereas in double stranded, the UV reading OD will be very high. It is because in a single stranded, you are exposing all the bases. Okay, whereas in double standard, all the base pairs are hydrogen bonded, so they are not that much exposed. So based on this UV difference, you can easily tell whether your DNA become a single stranded or like a double stranded. Okay, so I will send you this file, and there is another experiment that is for studying the secondary structure of your protein. So to find out the secondary structure, we have to go for CD, circular dichroism study. Okay, so if you are here, we might have done all these experiments in the lab. So unfortunately, we are not here. Okay, so that's why I'm giving you all these files. Okay, so circular dichroism, based on like your secondary structure, you will have like a like different graphs, and you can find out the secondary structure, the content of the secondary structure. You cannot say like whether the alpha helix or beta sheet is present in our like N-terminal region or middle portion or at the C-terminal region. That you cannot see. That you cannot say. But I can say like 40% alpha helix, 20% beta sheet, remaining helix. Like that, I can tell the content of secondary structure from the given protein. Okay, so to find out the secondary structure, we can use the CD spectra. Okay, so what I am doing is I will do. Go to the classroom. Okay, so I have sent like 3 files to you. Okay, in the name of like lab 2, lab 3, lab 4. So these files are like experiment files. So whatever that I have discussed to you so far. Okay, so and apart from that, I have given you demonstration from like VAST and daily server. So those server also you have to do like experiment and make it as like observation and make it like a record. Okay, so go through these files. So it is self-explanatory. I have given in like very detail. So if you have like any doubts, later on you can ask me. And yeah, so with this like I have taken like almost like most part of your syllabus from your from your practical portion. Okay, purification of protein and the crystallization of lysozyme. Okay, so that I cannot give you. So again, I have to give like only explanation only. Okay, so whatever that I have given to you. Okay, maybe I will take one more class about chromatography. Okay, purification of the protein. I will make one class in the next week about like chromatography and gel electrophoresis, so that you can have idea about like purification. Okay, so keeping this file, so get ready with your record because like we have very short time, and soon after next week, so you will have like exam fever, right? So before that, you have to wind up the record. So whatever thing that I have sent you the file and the remaining exercise, whatever that we have discussed, based on that, so all of you should prepare the record. And when will you submit? How long you will take? So next Monday, can I expect from all of you?
Yes, sir, a small doubt, sir. It should be handwritten, or we can make it in a Word file and send it to you?
You make it like a soft copy. Okay.
Okay, sir.
So for the servers like the daily and all those things, that you have to make your own. Okay, make your own statement. Don't take copy and paste from the server. All right. So you read that. Go to the daily server or vast server. You read the instruction. You prepare like files on your own. Okay.
Okay, sir.
Okay, so we stop
Sir,
here.
excuse me, sir.
Yeah, what's about?
Sir, yes, sir, I have a doubt, sir. Apart from this PDB, VAST, DALI, and everything we have doing, you have sent some documents, no, sir, around 4. We need some data for that, or else how can we document that 4 into our record, sir?
The dummy number, just put like dummy number.
Okay, we can take any number for this absorption and everything.
Yeah.
And we have to calculate.
You have to calculate using the formula that I have given.
Okay, sir. So table filling part is up to us.
Dummy number you are giving like 1, 2, 3, some random number, it will not work. If you understand properly, I have given like a different composition. Okay, so I have given like 1:90 ratio. Okay, 2:80 ratio, 3:70, something like that I have given in the table. If you go through that, I have given the like different composition. If you understand properly, okay, you will get like a proper answer. Okay, random you are giving and you are calculating, some number will come. Whether it will be correct or not, that you won't know. Okay, so try to understand the problem and give the reasonable numbers. All right.
Okay, sir.
So,
sir, and deadline?
Like I'm saying, like 1:9, 1:9 ratio means I'm taking like 1 amount of protein, 9 amount of like buffer. So 10 times I am diluting. So I am taking another reading, 2 amount of protein, 8 amount of water. Okay, the concentration is increasing. Yes or no? So next reading, I am doing like a 3 amount of protein, 7 amount of like water or like buffer. The concentration is still increasing. If the concentration is keep on increasing, the optical density, you should know what will happen. If the concentration is diluting, means what will be the optical density? So if you understand, if you have understanding on this, then only you will be able to like get like a proper curve, whatever. Okay, so try to work out and show it to me. Okay.
Yes, sir. Sir, shall we send next week, Wednesday or Thursday, sir? This week full we have test.
Next week, Wednesday you want?
Yes, sir. Wednesday or Thursday, if possible.
Okay, don't take more than that. Right?
Yes, sir. Is it Wednesday or Thursday we will send, sir?
Thursday.
Yes, sir.
Okay. Okay, guys. See you tomorrow. Okay, bye.
Thank you, sir.